Hello, everyone. For my final project, I decided to discuss human genome sequencing. Here's a little overview of the things that we'll talk about during this lecture. So the news article that I chose, some history about it, what genome sequencing is, how it's accomplished, and the importance of it. So the article that I chose is about human genome sequencing. The link there is provided there at the top. If you'd like to click on it and read about it, it is pretty interesting. But just an overview of it is scientists have deciphered the missing 8% of our genetic blueprint. That's a huge feat for us because now we can use it for other things in science that we'll discuss later on in this presentation. The first draft of a human genome was published in the early 2000s. Technology at that time could only produce 500 letters of our DNA at a time. And the human genome is about 3.2 billion letters long. So you can imagine why it took so long. It takes quite a bit when you can only do 500 letters at one time. This included about 100 researchers from government, private, and public organizations. Some history. In Module 5, we learned about genes and DNA, and kind of everyone's participation in this helped us get where we are today. So in 1944, Oswald Avery discovered that genes were made up of DNA. In 1950, Erwin Chargaff proposed the idea that DNA contains nucleotides. Nucleotides are letters present in a DNA sequence, A, T, C, and G, and we'll talk about that in just a few minutes. Chargaff noted that nucleotides are present in equal amounts, so there's the same amount of A's and T's as there are C and G's. In 1953, James Watson and Francis Crick discovered the double helix, or also known as the spiral staircase, and that's kind of what our DNA looks like. Chargaff discoveries helped Watson and Crick discover the double helix. So everyone's discoveries before their time was able to learn from that and help them with their discoveries. And now everyone all together has gotten us where we are today. So what is human genome sequencing? It's basically mapping out the human blueprint. It's how we are. So DNA is similar to a spiral staircase. You can see to the right there, you can see how it's the double helix. And then you see like the pink, orange, yellow, and green. So pink is the A's, orange is T's, green is C, and the yellows are G's. And A's and T's will always pair together, C and G's will always pair together. So basically what they're trying to do is figure out in which order all of these letters go for 3.2 billion letters long. It's quite a bit of a project. A, T, C, and G are the nitrogenous bases within a nucleotide. So there's 3 things that make up a nucleotide, and the nitrogenous base is a big part of it. So first genome sequencing was done in 1977 by Frederick Sanger. He developed the first DNA sequencing technique, which allowed for the first full genome to be sequenced. It was known as the Sanger sequence. It was a virus named bacteriophage FX174, and it contained 5,368 base pairs. Now, if you think about that compared to a human genome, where we have 3.2 letters long, obviously it was a significantly smaller scale, but it was still an impressive thing for science at the time. So first human genome sequencing technique is known as shotgun sequencing. It's done by Weber and Myers, developed this technique in 1997. By 1998, less than 5% of the genome had been sequencing. So it was a technique that took quite some time. And because of it taking a while, it was not well received by the science community. The goal of accomplishing the human genome by 2005 was uncertain due to how long it was taking. It involves randomly breaking up DNA into small fragments. Then those fragments are sequenced individually. A computer program then looks for overlaps in the fragments and reassembles them into the correct order to produce the genome. And it's kind of similar to how we do this today. So we obtain a DNA sample. Over there on the right, you can see a picture of a blood culture, and we're just going to follow the steps through. So you obtain a blood culture. You can then add chemicals to that, and it's going to release the DNA. Then the scientists slice the DNA into segments. Those segments are then made copies, and then they are uploaded into a sequencer. Specialized programs piece together the order in which the letters go, and you can see that on their the monitor of the computer screen. You can see how it's putting pieces together here and there. It's basically just like a huge puzzle that you have to see where those puzzle pieces fit. Finally, we got the importance of it. Like, why is this important? Well, we can have the ability to identify diseases in genetics and also identify genetic variations or mutations in genes and in our DNA. We can look for those mutations. For an example, in one of the peer-reviewed journals I read, an 11-month-old female diagnosed with hypercholesterolemia but she had a negative family history, was able to benefit from whole genome sequencing and find where she inherited the disease from. Her mom ended up having a recessive trait that she was unaware of. So that's kind of the importance of it, and that wraps up my presentation here. Thank you guys for listening.